All right, so let's start. Hi, I'm Fabrice Gorrec from Structural Studies at the LMB, and I'm a support scientist responsible for the Protein Crystallization Facility. The facility integrates 8 liquid handlers and other pieces of equipment. Historically, the main application was protein crystallization. However, there have been other applications supported occasionally, and today I will mention development of assays for biophysics and transcriptomics as well. The basis of automated liquid handling, well, first, it can be enabling, for example, to do very small drops, and we're going to see a system that can do that, 2.5 nanoliter drops, something like you can do by hand. More naturally, people think about the throughput, so it could be enabling high throughput. The rule of thumb is that the more simple and task you need to repeat, the more automation can be worth it. And what comes into mind as well is cost effectiveness. So this is based on different costs. We'll come back to that at the end of the talk. So before you're thinking about integrating an automated liquid handling system, there are a few considerations to think about. So what type of liquid you want to handle. The main culprit is the very low surface tension and volatile reagents. There's actually no system that can handle very low surface tension solution in the tip. So that's usually really a problem that you need to really anticipate. The volume range, we could split system into 2 categories, the one that can handle microliters and the one that can handle milliliters. No, sorry, the one that can handle nanoliters and the one that can handle microliters. Milliliters, there are systems that handle milliliters. That's more for industrial applications. And I'm not going to talk about those now. The precision you require, so there are things that need more or less precise. Usually you need to be very precise, especially nowadays that the scales of the experiments went down. So, you know, the thing is, how much do you want to pay for precision? That's a question as well that you need to anticipate. Cross-contamination, in general as well, you don't need cross-contamination. It depends, sometimes a very small cross-contamination is okay. If not, then you know, also you need to anticipate these two. Flexibility and complexity. So I put these notions together. So if you need to transfer a screen from 96 well played to 384 a couple of times a day, you may just want a semi-automated pipetting system with a 90/16 head, like the one pictured on the left there. And that's fine. That's all you need. If you need independent positioning with several tips, different type of dispensing, then maybe you need a fully integrated, and then you need very high throughput. Then you may want to design a fully automated system, like the one pictured on the bottom right corner there. It's a system that we developed at the LMB. We don't have it anymore, but it's a system integrating the Hamilton Lab Star, the Mosquito, and a sealer. But I'm not going to talk about that one today because we don't have it. Then you want to think about the speed. Is your protocol needs to be very fast because of evaporation, for example. That's also a consideration, or maybe it doesn't have to be fast, so maybe it will cost you less if it doesn't need to be very fast. Maybe there's an option for that. Cooling, so do you need your experiments to be at 4 degrees? Cooling units usually are quite expensive. The one that you integrate on your robot. There are also devices that you just pre-cool, like in aluminum. You put in the freeze, and then it's cool during the duration of your protocol, if your protocol is not too long. There's also robots that can work in the cold. So we have one of our Mosquitoes in the cold room, and it's working perfectly fine there. So that's also something you may want to ask yourself before implementing such equipment. And then the labware, Before the labware is just tubes and plates. Before there was, let's say, 10 plates available on the market. Now there's few hundred, so it's a bit of a headache, this part now. But it's very important to consider the details about this as well. So the plates for liquid handlers is usually called SBS plate for SBS format, which is basically a standard footprint dimensions, and also the well spacing, which will be 9 millimeter, which is the standard spacing between the tips of a liquid handler, or 4.5 for systems that have more higher density of tips. The manufacturing of the plates, it's important. So again, because there's a lot of plates now, but they are not all the same qualities. For example, if you have a plate which has lots of difference with the height of the well, where you need to dispense between one side to the other side of the plate, if you have half millimeter difference on the height, this can be a big problem later. If you do dispensing with touching tips at the bottom of the well, you're going to have problems. So watch out for the cheap plate. Sometimes there could be a problem. The properties of your plate. So do you need to be low bind, like maybe plates made of polypropylene? Do you need to be sterile, etc.? So how slick your plastic need to be as well is the same there. There are different manufacturing process and different quality on the surface of the plastic, which may or may not matter to your process, but you need to think about all those details. So here are the list of liquid handlers we have at the protein crystallization facility in the LMB. I listed them in different type. So by different type of pipetting, it's not always possible to compare them directly. The volume range, again, is something as well where you could compare them. So we have an EVO, and then the main advantage I've put as well, that's because I couldn't compare them directly. So I just thought about one of the main advantage on the top of my mind for each system. So we've got one EVO from a company called Tican, which I think is replaced by something called Fluent now. So this one is a liquid displacement system, and I'm going to explain that just after this slide. The volume range between 200 and 400 microliter. And then if I think about one main advantage of this system is that it has fixed tips, and I'm going to talk about that as well in a minute. We have 3 mosquitoes from the company SPT-Tech. This robot now is based on positive displacement for the liquid handling. It's for smaller volume, drop between 50 nanoliter and 1 microliter. According to who you're talking to, and especially manufacturer, there will be different minimum volumes announced. The mosquito can dispense smaller volume than that, but we don't need to go that low with that one. So I put 50 nanoliter. And then if I think about, again, the first thing top of my mind about main advantages of this system is that it doesn't need liquid classes. So because it's positive displacement, whatever the type of viscosity is, it will aspirate. You don't have to set up different settings for every different tips. This is not part of the picture. You don't have to worry about that. We have also 2 dragonflies from the same company. It's the same type of pipetting. It's not the same technology, but it's the same type of pipetting, which is positive displacement. It's for larger volumes. And the main consideration of that one is that it's very fast. And it was implemented initially for doing a lot of optimization screens. So we need the speed. We have a Labstar from Hamilton, which is now based on air displacement, which is basically what's used when you do a normal a manual pipette. And then the range of volume here is 0.5 microliter to 1 million. One thing come to my head with this system, it will be this notion of flexibility and complexity. It can do a lot of things invade in different ways. So that's really a very complete system. And then we integrated the echo liquid. So it's from the company. It was LabSite and it's now been bought by Beckham Coulter. It's not actually a pipetting system because it's acoustic ejection based. I'm going to explain that a bit later. And now we're going down to 2.5 nanoliter dispense. So, and that's the main advantage that I will put on the top of the list for the system. Just to mention that there are other type of system. A long time ago, we had a really cool system called the Cyclone from a company called Caliper. And it had a 96-channel head. And when every channel was independently controlled with pressure. So there was, this doesn't exist anymore. There is a robot that looks the same, that's called the same by Perkins, but that's not the same technology. So that was a really cool thing to have this independent control of pressure in a 96 head like that. But it's not made anymore. It was a bit pricey. And as far as I know, there's no equivalent. But I just wanted to mention that as a different type of pipetting for these machines. And then you may have heard about piezoelectric. So this one is about electric charge that is applied to the actuator that oscillates up and down at incredibly high rate of speed, generating kinetic energy. So you can dispense picoliter droplets with that one at very high frequency. So it can be very useful, but not really for our application because it can't handle all the type of liquids and especially not different type of liquids simultaneously. So we don't have really use for it. It's more for industrial applications. So liquid only based on liquid displacement. So when you see a water tank like the one at the bottom left corner there, well, you know it's a liquid only based on liquid displacement. So that's all the water that is needed during the process. I'm going to come back to that in a minute. So this is actually not only a liquid handler. So the part of the liquid handler is the EVO, but it's a fully automated system with other integrated pieces like a carousel on the right here, a sealer that is triggered through during the process automatically. And we had an inkjet printer as well to label the plates on the file. We don't have that anymore, but it was part of it before. So usually the liquid handlers based on liquid displacement usually have a multi-channel pump unit, usually with 8 tips. So basically that's the main mechanism for the liquid handling. So you have a 2-way channel valves. At one end you have the water tank, and on the other ends you have the tips. This kind of the water-based, the liquid displacement-based system are ideal for fixed tips that can be thoroughly flush. Nevertheless, even if you can flush the tips, you have eventually little reminder of the water in the tips, which can cause a very slight dilution over time in the process or cross-contamination. So not ideal for very strict cross-contamination matter. So we're using this fully automated system to transfer solution from tubes that we bought kits, protein crystallization kits in tubes to transfer it into plates. So we load the empty plates in the carousels. The gripper, forgot to mention, there's a gripper that transfer the plates onto the deck. The pipetting head transfer an 80 microliter of the conditions from tubes to the reservoir of the plates. And the plates is brought to the sealer. It's sealed and placed back in the carousel. So that's a very good system that is very reliable. We use for a very long time. It's very robust as well. So preparing a lot of protein crystallization crystallization screen in preloaded plate ready available to the users is our main strategy for crystallizing challenging samples, and also it gives the choices to the users to choose between different screens. So it's really an important part of the process. So we call the MRC plate field with crystallization screens the LMB plates. So, and we have 24 different screens now available. So we do produce still a lot of those. So once you have to proceed with protein crystallization experiments and other things, but protein crystallization is the main application again. We're using the mosquito. So now we are talking about a liquid handling based on positive displacement. And basically what it is a syringe, but like a micro syringe, because the piston is like a needle. And that's the basis of the technology. So we have 3 mosquito to dispense droplets of usually 100 nanoliter of protein plus 100 nanoliter of the condition from the crystallization reservoirs plate. So here is an example of 200 nanoliter protein crystallization droplets with protein crystals. I like to put this example because it shows all the different phases that you see on phase diagram with the clear and precipitates, microcrystals, and more the type of crystal you want. So that was just to show you an example because it's not, if you don't know about protein crystallization especially. So when you generate, so you do an high throughput protocols, and then you generate a lot of data. So you need a bit of help to analyze this data. For protein crystallization, we developed with collaboration with Paul Hart from the IT department, a web-based interface. So it's for basic data analysis, but it works really well, and multiple users can go and ask something in the database simultaneously from anywhere. So you just select the condition, and the database will show you the details of that condition. The next stage will be to think about implementing a LIMS system, a laboratory information management system. We don't really need that, and it's not something I'm going to discuss today. But other labs do integrate LIMS systems to analyze the data. So another word is the dragonfly, and I think I call it the metamorphosis of mosquito into dragonfly. It's because it's a bit like, it could be considered like a big mosquito. However, the syringe is not the same. It's not bigger because it's to handle larger volume, and we have a piston made of plastic here. That's the difference. So it's the same principle, and it could be compared to his predecessor, but for larger volume. It's quite, we actually developed this in collaboration with SPT LabTech initially some years ago. The reason is we wanted the speed to produce a lot of optimization screens directly into plates. And this gives us the speed. Not only it can handle viscous liquid because it's positive displacement, but it's also very fast because it's loading all the stock solution it needs initially in the big syringe, and after that it dispenses the whole thing. So that's why it's so fast. So the fundamental starting point for optimization of crystallization condition is to measure the effect of the concentration for the main components, ideally to produce better crystals, but also at least to reproduce crystals with your own stock solution. So for this, we use the 4-corner method. It's called 4-corner because the condition from 4 corners that will be found in 4 corners of the plates are prepared first manually. So that's easier to work at a larger scale, at a scale of milliliter in tubes. And the main reason why we do that is to have less, conditions to be handled that are less extreme. So they are diluted to start with. So that's the main optimization protocol we use based on the Dragonfly. The Dragonfly has from one, it's not 4 fixed tips, it's from 1 to 10 tips or syringes, should say. So there are other protocols that you can do with this, obviously. And some people, it's relatively easy to program as well, like the Mosquito. So there's more and more users doing their own custom programs. So the next system is air displacement system. And for that, this one is the Hamilton LabStar. The air displacement system is the principle of the manual pipettes that you use every day. The pipetting head of the LabStar is a bit more sophisticated than the pipette, though. And there's notably O-ring that enable to have really a very tightness when between the channel pipetting and the tip. This system is called the core system, and it also have a feature that where it can detect abnormal pressure. So, for example, if the tip is clogged, it will know because of the pressure built in the channel. So it's a bit more than a normal pipette. Then one of the optional features which I found really cool is called the core gripper, because it can connect to the same mechanism as the tip, but it's with using 2 of this head, you can it makes a gripper. So I found that quite interesting. There are other optional features like a more classic gripper. It's a bit complicated to program this one, but it can reach a high complexity of protocol, and it's also possible to have to build a fully automated system out of it. So it's quite good. So we're looking for we're investigating how to facilitate some protocols for single cell RNA sequencing in collaboration with Madeline Lancaster from Cell Biology Department and one of her team member, Hannah. One of these protocols is the normalization of concentration for cell samples. So it is actually a good old school protocol development with a liquid handler. We start with an Excel spreadsheets. So it can be only just you can just manually make a list on this spreadsheet, or you can do up to doing a prototype software with few macros where you can design the experiments, and the work list that you need for the liquid handler is automatically generated. So I'm still often working the same way as 20 years ago in doing that. That enables a lot of flexibility, because you can do a different Excel applet the next day if you need to. Then in general, the liquid handler, most liquid handler will can integrate a CSV file. So you have now a more basic list, and in this case, it will be a list of volume, the volume you need to dilute your sample and normalize the concentration. The cell samples are into a twin-tech semi-skirted plates that sits nicely into a pre-cooled aluminum block, and that's this that you're going to place on the deck of the robot. I would like to mention also the tips of the LabStar from Hamilton. They are made of polypropylene, and they give options about having a wild bore tips, which is a wider opening than normal at the end of the tips. That's very interesting for handling cells, both the fact that they're polypropylene and they have wild bore at the extremity. It's ideal for handling cells. Another investigation is how to facilitate the combinatorial barcoding method called SLIT-seq for split pool ligation-based transcriptome sequencing, using the PAR kits, which are able to you're able to proceed with 96 samples at the time. It's not 96 cells; it's 96 samples of cells. This method has several advantages that I will not discuss today, but I'm happy to discuss later. However, during this method, you have, because of the split and pool sequences, repetitively, you have a lot of multi-channel dispensing steps, like over 100. Plus, because you work with cells, you need to constantly mixing because the cells tend to settle at the bottom of the well. So this is definitely when the automated liquid only can enter in the picture to help. So we are looking into this. It's ready actually, but we're going to proceed with the first kit very soon. While I'm here, I'm taking the opportunity to mention the LM Institute, and here I'm with James and Karim Garbi doing my 3 weeks visits over there, where I've learned a lot of things. So thank you guys for everything. Right, the last type of system is not a dispensing system, it's an acoustic droplet ejection system. It's called the ECHO. So it's very cool technology, which basically you don't dispense anymore, you eject droplets. So this is the source plate. And with this acoustic transducer, it gives acoustic energy, and that's enough to eject the droplets to the destination plate, which is upside down. It's not a natural approach, but it does work. But yeah, you have to consider that your destination plate is upside down. So it has multiple advantages. It's very fast. You bypass the tips. It has automated. It can detect automatically as well the parameters required for different liquid classes. So that's cool. You bypass it, but you have to buy specific plates. The source plates need to have a very, very flat bottom, and they so you can't put any plates. So they sell specific plates for the ECHO. So you don't bypass the consumable there. So the main reason why we initially integrated this technology was for protein crystallization, because that's what we do mainly. And the idea was like it will require a bit less samples. So instead of 200 nanoliter droplet, here's the 20 nanometer droplets. And we made this work very nicely. And the idea was to make the protein crystallography more competitive, while less sample is required to proceed with protein crystallization. I don't think this is happening. It's not because of the technology. I'm not going to discuss the reason why it's not happening, but I'm happy to discuss this later. Fortunately, other people have found maybe more suited application for the ECHO, notably Jacob from the PNSE. He used it for affinity-based fluorescent polarization assay, sorry. So for this assay, measurements are based on light emitted by a fluorophore being polarized when it's bound to the macromolecule of interest or not, in which case it will still shine more. So with the speed and ability to eject small droplets, the ECHO enabled Jacob to proceed with initial screens containing 381 compounds in a 384 well ECHO plate. The source plate, there's 3 wells with DMSO only for control. And then he also programmed a dose response protocol, so where you have a serial dilution of the selected compounds. And he was very happy to have a possible range where you can go down to 2.5 nanoliter to build this range of concentration. It's getting very interesting, obviously, when you have expensive compound to work at with a lot smaller volumes. You save on the cost because of that. Right. So next, so in the bigger picture, programming is often the the limit of this for users. Even for me sometimes now. So there's people looking at solutions to facilitate programming. So there are already a lot of programs in cloud-based repositories like GitHub. There's people also talking about programming any robots with one single language, Python, is coming in the picture, which is quite a cool idea. And then AI is coming. Obviously, we don't know. Maybe there will be a robot, literally, program it for you. But we're not there yet. And in any case scenario, although there are a lot of tremendous development in the background with Python and AI, there are limitations here. So first, there's the evolution of the equipment and the controlling software and the operating system of your PC. This is always evolving, and there's often hiccup between if you upgrade your controlling software. What version do you have before? How fast this evolve? How does that fit with your operating system? What about the equipment? The equipment also now has more and more technology like inside. So it's also this technology is also upgraded regularly. How you do a very smart programming thing that consider all of that, and also lots of time is spent to optimize very fine adjustments on your protocol. So that's where I spend most of my time. That's what makes the difference between a protocol and the protocol, is that all the time you spend looking at the machine working, optimizing very small details to make a difference. And it's especially more true if you're talking about now smaller volume possibles. So that's actually, once you used to program the robot, this will be where you spend most time. And this is really a crafty part that you can't all anticipate and program. Also, making sense of a protocol in the bigger picture. So you want to, it's nice to program things, but you want it to be really useful in the bigger picture. And that's not something as well. That's something that really a good application specialist will think about. And last but not the least, as we say, the validity of the maintenance contract. So I talked about the companies about the programming a universal language and system to program their robots. They didn't especially answer. I think in the background, you obviously have a problem with the fact, like, if you use your own way of programming, which is different than the one supplied by the company, once the robot crash and breaks, I don't think they're ready to put that on the valid maintenance contract yet. So at the end, my thought is nothing beats quality hardware and experienced application specialists. If you don't have good hardware, you're going to spend a lot of time on the software trying to compensate for the weakness of your hardware. So, for example, if your tips are not very straight, do you want to develop a software that always consider this, or do you just want to make a nice hardware where the tip is always straight? I think you're winning if you do better hardware. And the specialist for all the considerations. A good specialist going in the picture is good as well for all the consideration I just mentioned in the previous slide. Talking about hardware, it's an opportunity to thank the extraordinary teams of Steve at our technical instrumentation workshop. And in fact, they did many parts for doing specific protocol that are parts that are not available on the market. They also do parts that are very expensive, available very expensive. So just for an example, if you remember earlier this cool gripper on the Hamilton Lab Star, it has two little pins there, which is just to grip the side of the plate. But we use that to make a SBS lid with a rubber inner seal. So it's something you put to avoid evaporation during the process if you have a lot of plates to be processed. And then we made it stockable, so that was good. And it was designed specifically for this gripper. So you have the two little holes there is for the pins of the gripper to go in. So that's one of the coolest customized parts made in a instrumentation workshop. That's the what's in the background. This is a pie chart of the yearly costs. So this is just to give a basis of this, because the different costs can vary more or less dramatically according to your choices initially. So fixed tips, for example, you will reduce your costs on tips. If you look at the consumables, between the tips, the trays, and the reagent, so the reagent as well, it can be more expensive. Depends what the reagent are directly dependent on what do you want from this, if you handle expensive compounds or cheap stock solution. But in general, you can see that the consumables are about half of your costs. So you need to think about this thoroughly before integrating a liquid handling system. Then I put a quarter for the equipment. So from the time you purchase every year, you have the cost of the equipment. Be aware as well that this cost is more and more about the software. So the cost of the software is just going higher and higher. So you need to take that in consideration. A good maintenance contract or not, it's up to you if you have expertise to deal with little problems in-house or not. We tend not to take maintenance contract and pay when there's a problem, pay for per visit. And then staff time, I would put that here too. It's good to have someone that handles this machine. It will make the things lots more efficient at the end. That's it. So I'm done for today, and I need to acknowledge Janlo and Olga Perisic. Those people are the founders of the protein crystallization platform 20 years ago, where it was not that usual to see robots in the lab. So those are the people that really put the basis of this at the LMB and developed the initial protein crystallization platform with robotics. And then also, I would like to thank the people from the operation groups, Christine, that helped to deal with general things and the budgets, Tom, who is always there to save me when I have a problem with IT, and there's also Poppy for the lab management in general, and Laura, who is helping me with the maintenance contract, did a great job with that. It's tricky to put acknowledgement because you can't put everybody. I could put the visual aids there as well to do some of the nice picture I use in this talk. And then I just mentioned the main company we're working with on a daily basis. And then with the application specialist, it can help as well when I'm stuck on certain development. And that's it for today. Any question?
I wanted to ask about that. When you were sonicating the droplets out of the reservoir, can you do that with can you like essentially emit a single droplet of the liquid you have in there, or does it have to be a few?
You're talking about the
sonicator.
The sonicator with 96 tips.
Yeah.
Yes.
It was no tips. They were the eco. Yeah, the eco.
The eco.
Yeah.
The eco. Yes. Okay. So that's this technology.
Yeah. Can you essentially emit a single droplet?
A single droplet. So the minimum unit of volume will be 2.5 nanoliter.
Mm-hmm.
So if you have one, so that's one ejection is 2.5 nanoliters. So that's the minimum volume as well.
Okay.
So and then you can do bigger with several ejection on the same spot.
For the sequencing applications, is there a magnetic block for if you had bead cleanup?
No, we're not there yet. So it's only, so you're talking about this kind of application. Yeah. No, so I'm looking only into the pipetting for now. So one thing at the time. I think it also depends. So you can the Hamilton Labstar is good for that too. It has option like some magnetic plates. You can even do PCR centrifuge. So there's almost all, so it has really all the optional features to develop protocol. Now it will depend on the protocol. The more you're adding steps, the more chance you have to have a hiccup on the way. I like to keep it more like a semi-automated protocol where you actually intervene, take the plates, check everything is okay. But you could think about more automated steps, yes. So we may want to do that in the future, but this is something that, you know, is just in progress. For now, it's only the pipetting. And I don't have shares in the Hamilton companies, but again, that's a good robot. There are other robots that can do these kind of things, but that's a good one for that. And maybe in the future, we're going to look into this. It's also about the motivation. So I'm in the structural study department. So this is the budget, and my budget is about protein, is for protein crystallization. So sometimes now, so now I'm stretching, you know, things a bit in different directions and try to find money from different groups to do more of this kind of things. But only the pipetting so far, for now. Okay.
Great. Thank you very much.